साड़ा है साड़ा होता है साड़ा क्या है आया आया बोलो देखिए नहीं है ये लक्ष्य है लक्ष्य नहीं
वो भी बैठा है सेक्सी बोल रहा है
बैठिए
हां वो लड़के लोग बैठे हैं सब लड़के बैठे
चिया टॉम बॉय
ना वैसे
नहीं हो
वही
टॉम बॉय
मर्दाना हरकत है
क्या
लड़की कम लड़का ज्यादा लगती है
तो
बोले थे ना उसको बायो का लेक्चर अटेंड करना
मैं टॉम बॉय कोई बकवास कर
अब अटेंड ही नहीं कर पाएगी वो
कौन
देख रहा है कोई तो उसको पूछेगा कौन थे
अनायता है शायद कौन है
वो क्या है जॉइंट
जो भी है
ओपन
वामोस
नो
और ये बंद कर
इधर वो तो वही है नो
वही है यू आर रिक्वेस्टेड टू लीव दिस स्ट्रीम राइट नाउ आपको इजाजत नहीं है ज्वाइन करने की फटफट जाओ
मैंने लीव किया उसने
यस आपको इजाजत नहीं है अटेंड करने की
माइक बंद कर
डाल ले अपने अंदर
वापस ज्वाइन किया ठीक है इसको बंद करो
भाई बंद करो
रुक ये अनायता है तो लाइक कर
अगर चाहो तो लाइक कर
तो ही रखेंगे स्ट्रीम मैं तो बंद कर देंगे
मैं तो नाम को बोल के बंद करा दूंगा
यूट्यूब देख के लाइक नहीं करते
दो लोग कैसे देख रहे हैं अभी
किसको बुलाया तूने
किसको बुलाया इधर दिस इज माय क्लास कम दिस इज माय क्लास कह दे लो सीनियर का स्टडी था
ए लाइक किया और कौन है
गूगल से क्या है ऐसा क्या है अच्छा से गाने जाता
क्या है
यो कौन ओ
पहली बार थी
क्या बोलो ना ये बोलो किसने लगाई बोला मुझे लगा था तुझे ही बोला
कहाँ रतन की रही है
देखो पानी का देखो बॉटल दे पानी के लिए
उसमें से पी लेगा मैं देख रही अरे क्या आ जा रही
लाइक प्लीज डिसलाइक करके लाइक करिए वापस
आ रही है तो उसका नाम लिख दे शी इज एब्सेंट
इसकी स्पेलिंग नहीं आती मेरे को
ANAHITA
रुक जा ये लिख दो
हाँ बोलिए हाँ लिख अरे ट्रायंगल बनाना करने से नहीं ट्रायंगल बनाते हाँ अरे AJ ट्रायंगल बना दे इधर लिख proportional to
नया एंड
नहीं 1/Ganesh
नहीं अरे यार लिख नहीं तो करना ना कर एंड फुल सपोर्ट है आपको
बहुत तो ये खड़ा रहते ये लेके रख के Kanish. Kanish. True. Times.
No one. Right, can't do it. वो अभी चली गई, वो अभी मैसेज कर रही होगी व्हाट्सएप पे।
हाँ, वो मेरे को ये बोला।
मैं बस ओके आई जॉइन। कोई नहीं, मैं मार डालूंगी। Do we care? Do we care? No, we don't.
स्टिकर भेजो क्या इसको?
भेज। डरते क्या? आज देख 2 दिन तो बच जाएंगे अब, ठीक है?
अरे नहीं, बच्चों को
अबे यार पागल क्या है, इतना डर रहा है।
सुविधा का चेहरा दिखाऊंगा।
क्या?
सुविधा का चेहरा दिखाऊंगा।
ना भाई की डिनर करके आएगी ना।
अपना इसमें से किसी का लगा? अपना इसमें से किसके ने अपना ये लगा है?
पीएसपी।
हाँ, प्रोफाइल। ये कौन है?
प्रोफाइल पे की बोलते? एक वो है कनिष्का। दिखाने की कोई चीज कोई है नहीं। मेरा मत दिखाओ। नहीं मेरा क्या है?
इधर है नहीं ना। ये कौन है?
ये तो
नंदनी।
आई गेस हाँ नंदनी है।
This is Nandani. This is I don't know. Kune. Who is this? No idea.
हाँ दिखा?
कौन है?
ये देख मेरे को तो अलग ही दिखा रहा है। अच्छा
पता नहीं भाई।
This is bullshit. मामा
That's me. देख।
नहीं that's me थोड़ी।
आपको इससे लिखा।
Only both of you today.
हाँ, आपको ऑनलाइन।
5 people out from.
कौन है ऑनलाइन?
हाँ नहीं सर, लीव कर दिया उसने।
अभी 2 मिनट पहले थी चाहिए। हम कोई बच्चा नहीं है।
हम एक ही लेक्चर रखते हैं।
कोई नहीं आया।
I know फिल्म के लिए फिर से रिपीट करना पड़ेगा।
हम ऑनलाइन स्टॉप चल रहा है तब से।
तुम बैठो पहले।
I was telling you that.
Okay, see here. Which was the chapter we were doing?
Structure of chromosomes.
Structure of chromosomes and cell division, no? Chalo, we'll complete with cell division. Textbook, both of you don't have? Give me one book. एक book इधर है. Let me see. The next part is. Okay, so we completed with the process of cell division. Cell division समझ में आया? What are the stages of cell division we learned? Mitosis. What are the features of mitosis? Tell me.
Mitosis.
Features of mitosis. What are some of the features of mitosis? Number 1, mitotic cell division takes place in what type of cells? Somatic cells. Phone rakho andar. Takes place in somatic cells. How many daughter cells are formed?
2.
What about the chromosome number?
Same.
Maintained. Chromosome number maintained. What about the characteristics of the offsprings?
Yeah.
Identical to the parents. What are the basic stages? What are the main stages of cell cycle?
Including interface or?
For cell cycle, you don't have to write interface.
Metaphase.
This is all interface only. Before mitosis, what you are talking about, G1, S, and G2. This is all interface only. So, what are the stages of cell cycle?
Prophase, metaphase, anaphase, telophase, and cytokinesis.
Prophase, metaphase, anaphase, telophase, cytokinesis. These are the stages of mitosis. I asked you stages of cell cycle.
Cell cycle.
S phase, I'm sorry, G1 phase,
S phase, then G2.
G2, and then M. So first is G1, S, G2, followed by mitosis. Major difference between G1 and G2? G1 is a longer phase. G1 is a much longer phase than G2. In the G1 phase, the cell becomes larger, develops all the cell organelles, proteins, RNA. G2 is a comparatively smaller phase, only prepares the cell for division. G2 is just the finishing phase, preparing the cell for division so that any additional proteins, enzymes, factors necessary, so that has to be formed. Clear? Now, you will be asked questions, what is the significance of mitosis? What do you write there? What is the significance of mitosis? So if we talk about the concept mitosis, and now today we are going to compare with the concept of meiosis also and learn certain few terms. Significance, I start with this. First is significance to understand. What is the significance of meiosis, mitosis? I'm talking about mitosis first. What is the significance of mitosis? Again, the same things.
The chromosome number is
Maintained in the daughter cells. Chromosome numbers are maintained in the daughter cells. That is the significance. And number two,
parental
identity. Parental characteristics, or the offsprings are identical to the parents. So now, when we talk about the stages of plant mitosis, what were the differences we learned?
formation of first of all, there is no centriole and centrosome. Then there is the formation of cell plate instead of the formation of a furrow in anaphase.
In the anaphase. So what are the primary differences between plant and animal mitosis? Number one, in the anaphase stage, or I start from the metaphase stage. In the metaphase stage, the spindle is formed without the centriole. I talk about the anaphase. In the anaphase, if it is the plant cell, cell plate is formed without furrow formation, no furrow formation, only the cell plate. And in the telophase, cell wall is also formed. In the telophase, after the process of cytoplasmic division, telophase, when it is the time of cytoplasmic division also, just at the end of the cytoplasmic division, the cell plate will also be formed apart, the cell wall will also be formed apart from the cell membrane. So these are the differences that you have to remember, plant and animal mitosis. Did we draw certain few diagrams? All the stages of animal cells. Comparatively, remember the stages of plant cells also. Plant cells also we have done?
Plant cells we have drawn metaphase.
Some questions here I think we have already solved. Which are the questions we have solved? Now today we are going to complete with meiosis. But which are the questions we have solved? Let us complete with the questions first. Just one or two questions left for meiosis. Which are the questions we have completed? Which are the questions? Now the MCQ question. Okay, now let us complete with meiosis. Then at one go you will complete with all the questions. That's much better. We now talk about meiosis. Now let us compare. What about mitosis and what's new about meiosis? I already the other day also, when I was talking about the concept of mitosis, there also I mentioned some differences between mitosis and meiosis. If it is a concept of meiosis, which type of cells will be undergoing the process of meiosis? It is always the reproductive cells. Cells of the reproductive organs only will undergo the process of meiosis and result in the formation of specialized germ cells known as gametes. What is the second feature to understand? How many daughter cells formed? In case of mitosis, we have understood 2, and now when it is meiosis, we have to understand 4 daughter cells always formed. So for the features concerned, when we are talking about In case of mitosis, the parental characteristics are retained. In case of meiosis, always we have to say that the variations are there. New characteristics are always formed. Now, I try understanding one particular cell in meiosis. How would be the basic stages? I am talking about, I start with only one chromosome here to understand. This is one particular cell, one chromosome. Okay. Now, this particular chromosome, what will happen if it is mitosis and prophase stage? What will happen if it is the prophase stage? Or let me talk about a cell with two chromosomes, easier for you to understand. I talk about a cell, I remember I'm starting with a cell with two chromosomes. This is the parent cell I'm starting with. Now, if this is the parent cell, and if I have to talk about a concept of prophase now, I'm only drawing the nuclear region. If I have to talk about the prophase, how will I draw?
The chromosomes will be joined at the centromere.
That means, what do you call this as? Chromosome duplication. Chromosome duplication, DNA duplication. So here I would draw something like this. But let me make it even more clear this way. I am drawing the two chromosomes this way now. This is one particular parent cell which has got two chromosomes. Now here I am saying it has got two chromosomes, means what? The diploid number is 2. What is the meaning of the diploid number? I mentioned the other day also. Two half sets of chromosome. Two half sets. Meaning this is one, I talk about this is one half set, this is another half set. What is the meaning of half set? Half set means whether when we are talking about in our condition also the chromosomes that pair up. Okay. So I am talking about this particular cell which has got two chromosomes, but when I understand, even in our cells also, say suppose, I am talking about in our cells, what is the condition? In our cells, I would understand this is 1 and this is 2 and this is 3 and 4 and 5 and 6. So what is the chromosome number I am saying? For example, what is the diploid number you will say? 6. Yes or no? 6 chromosomes, no? Now, every 6 chromosome, when I say, the chromosomes are actually paired up like this. So, if 2n is the diploid number, we say the diploid number is 6. And what is the haploid number?
3.
3. I talk about the definition again. What is the meaning of the word haploid and diploid? Haploid means one half set of the chromosome. If I talk about the blue chromosomes, 1, 2, 3, haploid. Diploid, when they are paired. So again, talk about the same concept here. Are the chromosomes in our cells single like this? No, actually they are always paired. So here, first of all, I understand that these two chromosomes, if it is meiosis, now we have to understand this better, that these two chromosomes, they will always pair up like this first. These two chromosomes, as I draw there also, these two chromosomes, meaning which two chromosomes? One chromosome that we get from the mother and the other chromosome that we get from the father. In our cells also, always the chromosomes are like this arranged in pairs. Then what are these? What is this chromosome? We got this from our mother. What about this chromosome? We got this from our father. We say this is the maternal and this is the paternal. What is the meaning? The maternal chromosome we get from the mother, paternal we get from the father. Then what is this pair of chromosome known as? This is what is known as a homologous pair. What is this known as? This is known as the homologous pair of chromosome. Understood? In our cells, every time the chromosomes are paired, we always say how many pairs? 23 pairs. Every pair of these 23, every pair, that we talk about is a homologous pair. Then what is the meaning of this homologous pair? One is a maternal chromosome, the other is a paternal chromosome. Understanding? Now we try the concept of prophase. Now if I have to talk about, now I proceed further. Now if I have to talk about the process of the concept of prophase, but this is meiosis, prophase and metaphase, the entire phase, the entire concept of cell division has two phases. So I am writing it as prophase 1, the first phase. Now in the first phase, after the pairing, then after that will be the duplication. The duplication is not the stage before. So first is the pairing that happens. And now the duplication. Now if the duplication happens, then the duplication is something like this. This is one. And this also is duplicating. But they are quite close. I'm talking about only one pair now. Two chromosomes forming a pair. So now these two chromosomes, what did we name it as? Homologous pair. I already told you what is the meaning of homologous pair. Now they are duplicating. So in the prophase stage, this is a situation. How would they behave in the metaphase? How should they behave in the metaphase? What is the feature of a metaphasic cell?
They will get joined at the center.
They will, metaphase, they will all align in the center. Metaphasic plate. This is an animal cell I consider. So if this is an animal cell and then the centrosome and the centromere, all these things are there, followed by that the spindle is there. Now I talk about the chromosome. How will the chromosome behave? This is one pair. And where is the other pair? The other pair will be very close because they are in pairs. This is how they will arrange themselves in the metaphasic plate. What was the difference in the mitotic stages we had understood? Only one pair was there. We did not talk about two pairs like this, no? Okay, I mean after duplication. Now next is what? Next is I have to understand then how do they behave during anaphase? How will they behave during anaphase? Okay, this centrosome, centromere remains as it is. We are talking about the spindle. As we talk about the spindle, now next one, how will these chromosomes get separated? Metaphase followed by anaphase. In anaphase, always the separation has to happen. But here, in case of anaphase, can you tell me what was one special feature of anaphase in mitosis we studied?
For the shortening of the spindle.
Okay, and?
Pulling off the sister chromatids towards opposite poles.
Okay, pulling off the sister chromatids, meaning I can also say splitting of the centromere. Splitting of the centromere. What did we learn in anaphasic stage of mitosis? In mitosis, always we had learned that this pair of chromosomes splits up. One goes this side and the other comes this side. But if it is the prophase 1 and anaphase 1, there is no centromeric division at all. There is no centromeric division at all. Clear? Now, before the centromeric division, we can talk about, there is something more to understand here. So, first of all, we understand the pairing this side and then the duplication. Before they start their separation, something more happens. What is it? This particular chromosome, yes, this particular chromosome is this way, one pair is this. What about the other one? The other pair is arranged in this manner. The other pair is arranged in this manner. What is this particular stage called? This stage is called the tetrad. This is called tetrad. Why? There are 4 chromatids. What will we call this as? These are known as the chromosome arms or the chromatid. So in the tetrad stage, what is happening in meiosis? This is an exceptional behavior of the chromosomes. What happens? These 2 duplicated chromosomes, they will align side by side. What is this aligning itself known as? This aligning itself is known as synapses. Coming together and aligning side by side. Why they are aligning side by side? Because one is a maternal chromosome, one is a paternal chromosome, and the sequence of genes are similar. That's why. Again, to understand, can I believe that this is the 9th chromosome and this is the 6th chromosome? Is this possible? No. It has to be the 6th and the 6th, or the 9th and the 9th, or the 12th and the 12th, or the 22nd and the 22nd, always like this. The maternal and the paternal chromosome, the 6th and the 6th, the 2nd and the 2nd, the 5th and the 5th, the 9th and the 9th, always like this. Why? Gene sequences are the same. For the 9th and the 6th, the gene sequence will not be the same, no? Now that they come together, now next one, after this, what is this type of a configuration known as X-shaped configuration? So what will we call this process as? This process is known as crossing over or cross joining. This is called crossing over or cross joining. After cross joining, what next? After cross joining is exchange of chromosomal segments. Exchange of chromosomal segments taking place. Exchange of chromosomal segments. Then, if this is so, then how should the chromosome look like now? I say that this is one particular chromosome, and now what has happened? A part of this chromosome got exchanged. A part of this chromosome got exchanged. Clear? So this is exchange of chromosomes that we understand. Now they got split up. What now next? This is one chromosome, and what will happen to this? A part of the chromosome got exchanged like this. So this is what we are always supposed to understand. Now, what is the reason? We had already talked about this. Variation. Variation. Why does variation happen? Because of this process. What is this process? Exchange of chromosome. Because of the cross joining, because of the crossing over, exchange of chromosomes happen. Our parents' chromosomes were not like this, but during the process of crossing over and cell division, this has happened. So our chromosomes are like this, and this process of crossing over is giving rise to what? New characteristics that we show. What is this set of new characteristics known as variation? Is this clear? Now we continue with this. Then what about this set of chromosomes? So we have to understand that these chromosomes are something like this. These are the exchanged chromosomes. These are the chromosomes with the exchange segments. Yes or no? After the splitting. So I have drawn first of all that they were aligned in the metaphasic plane, but by that time they have gone through this process. Now they are separated. Now that they are separated, they are aligned like this. Now there are names for each of those stages. So now that they are separated, and it is already the anaphase stage, how will these chromosomes behave? Is there any centromeric division? No, there is no centromeric division in anaphase 1. Then how will these chromosomes behave? This chromosome, they will be pulled like this. One chromosome is this way. This is the first chromosome that we are talking about. We are talking about the differences. What about the other side? If this is the other chromosome, we understand this is the other chromosome where these are the exchange segments. Clear? So this recombinants, or these are also known as the recombinants. What is the meaning of recombinants? Where the chromosomes have already exchanged their segments. Now they are moving like this. And after some time, now that anaphase has happened, yes. Will the furrow formation happen in anaphase 1? Yes. What now next? This will give rise to, after anaphase 1, telophase 1, cytokinesis. So after this process of cytokinesis, if I say, after anaphase 1, now telophase 1 will also happen. And after telophase 1, then cytokinesis happening. Then after cytokinesis, what is the feature of the two cells? Now I get two cells here. Now I am getting 2 cells here. Cytokinase is almost over. Now I get 2 cells here. This is the centrosome, centromere, and how are the chromosomes arranged? These are the chromosomes arranged. These are the chromosomes arranged that I come across. But this chromosome has got the exchange segments. These chromosomes have got the exchange segments. 2 cells formed. Now next one, this 2 cells. Now how will they behave? They will again go on with the same process that we had come across in mitosis. So now if I go again, that this cell, how many chromosomes? 2 already. This chromosome already 2. And now if it is the concept of prophase. Now it is the concept of prophase. Are they duplicating again? No, they are not duplicating again. Now in this case, they are just arranging themselves. Now they are not duplicating further and followed by metaphase. Now if it is the metaphase stage, meaning I am talking about metaphase 2 here. This is the metaphase 2 stage. This is metaphase 2 now. If it is a concept of metaphase 2, then this particular cell will arrange in the metaphasic plate something like this. Arrange in the metaphasic plate something like this. I talk about one cell. The behavior of this cell also will be the same. Then, if this is arranging in the metaphasic plate and followed by, if this is arranging in the metaphasic plate and they are arranging exactly in the equator, exactly this way, then what will be the next feature? And if it is anaphase and followed by telophase. I skipped one stage and I now straight away talk about the telophase. And if I am again talking about the telophase, from this cell, how many cells are we supposed to get? 2 new cells. Talk about telophase. How many chromosomes am I talking about here? 1 and 1. But the difference is this chromosome has got a recombinant, and I talk about the 2 more cells from here, 1 and 1. And what about the chromosome? 1 here and 1 here, and 1 has got a recombinant. How many daughter cells do we get if we follow the process? 4 cells. We are getting 4 cells. So how many chromosomes? What is the chromosome number in each of them? 1. We started with the parent cell with 2. What are we going to conclude now? Is the chromosome number reduced to half? Yes, the chromosome number is reduced to half. We started with the parent cell which had diploid number 2. Now, which are these cells we are talking about? We are talking about the cells of the testis. We are talking about the cells of the ovary. Only for animals? No, also for the plants. For the plants, then what? We are talking about the cells of the anther and we are talking about the cells of the ovary. So this is for the plant reproductive organs, this is for the animal reproductive organs. So the concept of meiosis in the plant and the animal will be absolutely the same, and the differences in the plant and the animal cell also remains the same. What are the differences in the plant and the animal cells? Again, no centrosome, no furrow, no cell wall. So those differences in case of meiosis also remains the same. But the features of meiosis, now that I talk about the 2 elaborate phases, meiosis 1, meiosis 2 in the plants also, yes. I talk about the reduction of the chromosome number to half, yes. I talk about meiosis taking place in the reproductive, in the cells of the reproductive organs, yes. So all these features remain exactly the same whenever we are talking about the concept of meiosis. Now, what are some additional terms you had come across in case of meiosis? You had come across what is known as homologous pair. You are asked definitions. What will you write? Homologous pair, a pair of chromosomes which consists of the maternal and the paternal homologue, or the maternal and the paternal chromosome which align side by side. What is this process of aligning side by side? It's known as synapsis. What is this condition known as when the homologous chromosomes are in their paired condition and overlap chromosomes? The tetrad condition. What happens after the tetrad, or what is happening in this process when they are overlapping, form a X-shaped configuration? This is known as, the process is known as crossing over. But what is this X-shaped configuration known as? This X-shaped configuration, if I say, this X-shaped configuration is known as chiasma. The X-shaped configuration that forms is known as chiasma. Process is known as crossing over. What does crossing over give rise to? Exchange of chromosomal segments. Clear? Exchange of chromosomal segments. And exchange of chromosomal segments will lead to variation. So only this much for meiosis. The stage diagrams and drawings you don't have to study at all. Significance of meiosis, see there, the basic phases. This set of diagrams, if you check, the set of diagrams in page 17, check there. The set of diagrams in page 17, this set of diagrams for identification only may come, diagram drawing will not come at all. Okay, it is not in syllabus, but in the paper exam, in the exam paper, this especially these stages of, see there, the stages of first division, second division, generally the metaphase and the anaphase stages only are asked. And you will be asked to mark this, either this or what is meant by this pair of a chromosome this way. Go to the next, there is one more diagram on the homologous chromosome. Where is it? There was one more diagram on the homologous chromosome. See there, in the set of questions it is mentioned. This is also asked. See there, page 22. Page 22. Look at the pair of chromosomes, the homologous pair. Did you see that? This pair of chromosomes and the process and exchange of chromosomes. So this is what is asked. Try this question. Tell me if it is understood. Look into page 21 now. This page 21, this set of questions, did you complete? Let us start from define. The define part, did you complete? Page 21, define part, did you complete? No, we have not done the set of questions. Define part we haven't completed. Give reason also we did not do. We have not done. Page 21 itself. We haven't done. Then we'll have to finish up with this. Let me complete with this set of questions and the ones from page 20 you will do on your own. Okay, continue. Write it in a notebook. Page 21. Page 21. Only few to write, not all. Chromosomes, how will you define? Chromosomes, how will you define? Are you aware about your school results?
शायद अरे मतलब मेरे नेबर को 91 आया
तेरा पार्टनर
यजत आता था ना यहाँ पे
यजत हाँ
91%.
Yajat, yes, I remember. But what about that? I did not see that. But your school has done superb results. Superb results.
और कौन आया था उनको उधर से?
यहाँ पे there were three more were there. That girl, I forgot her name. One more boy was there. One more boy was there. Rekha ma'am would know. One more boy was there. Another girl was there. हाँ, यजत का भी याद आ रहा है मुझे.
यजत कुमार. नहीं, तो अपने स्कूल का है.
Okay, come on. Write this. Define number 1 chromosomes. Chromosomes you know how to define. How could you define chromosomes?
Condensed chromatin
fiber. Extensively condensed chromatin fiber. Extensively condensed. Gene, you know the definition. What is it? Gene, what would you write?
Contains parental characteristics.
What is the definition of gene?
It's contained in DNA.
Functional segments of the DNA molecule. Write down some of these important ones. Write down the answers nicely. Gene. What is the definition? It is a functional segment of the DNA molecule that encodes for a protein. Right here. Right. Definition of gene. What is the definition of gene that you should write? Gene, functional segment. Is it not a functional segment of the DNA? How did we learn? What was the DNA? If I talk about that, this is the DNA molecule. If I say that this is the DNA molecule, which part is the
functional
segment of a DNA molecule
that encodes the protein and is responsible for regulating
a character? Exactly. That is the definition. Always you should write. What are these repeating units? What are these repeating units? Huh?
Nucleotides.
Repeating units are nucleotides. And what are these nucleotides made up of?
3 parts: sugar, pentose sugar, nitrogenous base, and phosphate.
And phosphate group. What are the 4 types of nitrogenous bases?
4 types of
Nitrogenous bases. Adenine, guanine, pado, cytosine, thymine, adenine, guanine, cytosine, thymine. 4 types of nitrogenous bases, and they fall in 2 categories. What is common? Adenine, guanine, they are known as purines. Cytosine, thymine, they are known as pyrimidines. Okay, these words you have to remember. Okay, now definition of the gene. What to write? Definition of the gene is functional segments of the DNA that encodes a protein that regulates a specific character. Then cell division, you should be able to understand. What is the definition of cell division? You will write definition. अगर पूछा है क्या लिखना है cell division? Process of formation of new cells or daughter cells from the parental cell. Process of formation of daughter cells from parental cells. The process of formation of daughter cells from parental cells is known as cell division. Process of formation of daughter cells from parental cells is known as cell division. Chromatid, what would you write?
Arm of a chromosome.
Arm of a chromosome consisting of extensively coiled chromatin fibers. Chromatid definition is always arms of the chromosome consisting of extensively coiled chromatin threads. Ester, definition of ester. Fine protein. What are esters? Remember those esters? Where do we draw these esters? Say suppose if I draw this properly, then this is the centromere, and here we draw these esters. These are the esters or the estral rays. Esters or the estral rays. So now that we talk about the esters or estral rays, what is it? This is the fine protein fibers developed from or produced by centrioles. Protein fibers produced by centrioles, they are produced by the centriole itself. Protein fibers produced by centrioles. Your school has already finished with genetics, you said?
Genetics.
Or before?
No, not genetics is going on.
Going on. And going on means you all are not having any extra class right now?
No.
Have we? No. And in the 9th, did they finish any chapter?
What time? No.
Nothing they have finished. Okay, then, okay, so this is about the concept of genes. Esters. Give reason. The first one, see there. Gametes must be produced by meiosis for sexual reproduction. Gametes must be produced by meiosis. What is the meaning? Gametes must be produced by meiosis. The major concept of meiosis here is Gametes should always have half the chromosome number.
Because while forming a zygote, the chromosome number will automatically become 2n.
So if the gametes would have been formed by mitosis, then what?
Then the chromosome number would have become 4n.
Then the chromosome number would have doubled. And if the chromosome number is more, the cell will not be able to function, because more the chromosome and the cell apparatus, that is what we call it as a cell apparatus, meaning the cytoplasm and the organelles, they will not be able to function properly. And on top of that, the characteristics of the parents and the offspring will be absolutely different. A complete whole set of chromosome. Remember, whenever we are talking about chromosome abnormality, rare conditions, certain children are born with one chromosome extra. Rare conditions I'm talking about. Certain children may be born with one chromosome less. One chromosome less or a part of the chromosome not there, that itself is enough to make the entire individual absolutely abnormal in various different ways. And here we are talking about a presence of excess or complete set of chromosome in double dose. So you can imagine the organism will be absolutely a different one. So what is the answer that you should write? Gametes must be produced by meiosis for sexual reproduction. Write down that answer. Gametes produced by meiosis result in haploid set of chromosomes. Gametes produced by meiosis result in haploid set of chromosomes, comma, which during fertilization restore the chromosome number, which during fertilization restore the chromosome number, which during fertilization restore the chromosome number of that organism. Formation of gametes by mitosis would result in presence of an extra complete set of chromosomes and may result in the formation of completely different group of organisms. Completely different group of organisms. Read number B. What should the answer be?
Because the chromosome number is reduced
to half. Reduced to half. But now tell me, when is the chromosome number reduced to half? In the first phase or the second phase? I'm just asking you this. Not there in the question. In the second phase. So now when we are talking about why is meiosis referred to as reductional division? Your answer always is meiosis is known as reductional division because the chromosome number is always reduced to half. Read the next. The children of same parents, however similar, are different from each other in certain aspects because of variations. So say suppose if you have your siblings, you both are from the same parents, but both of you, do you have the same set of characteristics? No. Why? Because the changes in the chromosomes in your body and his or her body is not the same. So this gives us variations. So your answer would be, how do you frame your answer? The differences are because of the variations which are caused due to crossing over and exchange of chromosomal segments. So this word has to be mentioned. Distinguish between. This is very simple. Cytokinesis and karyokinesis. What to write?
Division of cytoplasm and division of nucleus.
Nucleus. The stages of nuclear division, karyokinesis, cytokinesis, the stages of, or cytokinesis just one phase, the division of cytoplasm. DNA, RNA, distinguish.
DNA, deoxyribonucleic.
Distinguish the following pairs. Now they have not mentioned any feature. DNA, RNA, you can differentiate based on the composition, based on the organisms present, etc., etc. Now you would majorly get full form. You would majorly get full form or the composition, nothing else. DNA, what is the difference in the DNA, RNA? Difference in the full form?
Deoxy. DNA.
Big name, DNA, RNA difference?
Deoxyribonucleic acid. It has two oxygen molecules.
नहीं, composition लिखोगे क्या? आंसर में क्या लिखोगे? DNA and RNA composition अगर आंसर में लिखना है, क्या लिखना है?
That DNA does not contain uracil as a nitrogenous base.
DNA, the composition of DNA is nitrogenous bases, pentose sugar and phosphate group, and the nitrogenous bases are adenine, guanine, cytosine and thymine. RNA also has got nitrogenous bases, pentose sugar and phosphate group, and the nitrogenous bases are adenine, guanine, cytosine and uracil. Thymine नहीं है, thymine की जगह uracil. DNA and RNA composition हो गया, and is there any other difference? DNA and RNA. DNA is double stranded, RNA is single stranded. Write down this. Write this answer. This is there in your book, but you may not be able to frame it properly. DNA, RNA लिखो. Two differences, right? Every chapter, now remember, now that you are in the 10th, and if you have a goal, कितना मिलने से खुश रहोगे? हाँ?
75 plus.
कल से मत आना यहाँ पे. 70 तो तुम घर पे बैठ के भी कर सकते हो.
75 plus.
76.
हाँ 76.
मिलने से खुश?
और तो थोड़ा टफ है ना 80 का.
पर्सन ही हो, सिर्फ बायो को बोला.
हाँ?
सिर्फ बायो को बोला.
बायो का ही मैं पूछ रही हूँ. तो बाकी सब्जेक्ट्स में कितना?
बाकी मैथ्स 80, फिजिक्स पता नहीं, फिजिक्स 77, केमिस्ट्री 75 प्लस, लिटरेचर 80, हिस्ट्री 70, ज्योग्राफी 65, इंग्लिश लैंग्वेज भी 65.
लैंग्वेज
हाँ मैम मार्क्स कितना करते हैं? आज से 70 नहीं मिला है लैंग्वेज में.
What are you talking about?
देखो पहला 70 आज से लैंग्वेज.
What is the highest mark language then in your class?
72. 72. Our teacher said that she would cut at least 8 marks in letter. Then highest you can get is 72 only. Out of that I got 67.
Language में 72, 75 आता ही है. Now it depends how you all write. I don't know. I'm not commenting anything on that. Now talking about the science and the math subjects, if you have a target of getting only, how much you said? 70 you said?
75 plus.
हाँ.
तो उससे नीचे नहीं आना चाहिए. अच्छा है लगता है. 75 के नीचे आएगा. Because if all subjects are above 75, then I can get 90 plus.
Of course. 77, 78 तो मिलना ही चाहिए. At the most 1-2 mark का error, it's okay. At the most 1-2 marks का error, it's okay. So for that, now see, it's not very easy, huh? But what I'm telling you is, it's not very easy to get that 77, 78. Every particular chapter you have to revise minimum 5 to 8 times. Every particular chapter. मतलब चैप्टर के लगभग सब कुछ तुम्हें समझ में आ जाना चाहिए. अब इसमें भी एक आध चैप्टर यू विल नॉट स्टडी वेल, बायो हो, फिजिक्स हो, केमिस्ट्री हो, इसमें एक दो चैप्टर रहेगा विच यू आर नॉट वेरी कंफर्टेबल विद, जो तुम छोड़ोगे. So taking all that in consideration, तुम्हारा error should not be more than 2 to 3 marks. I'll tell you, my students, they used to, when I used to correct their papers, my school, I'm not talking about your school. When I correct their papers, now these are the students who got 99.6 percentage. 99.6, 99.8, both the children got. And when I used to correct their papers, every exam, even in the pre-boards, 1 mark error. वो भी 1 mark में जानबूझ के ढूंढ के 1-2 mark में कैंसिल कर दिया. Otherwise it was just errorless. मतलब when I am correcting those papers, I have to be extra with those binoculars कि कहाँ पे defect है ढूंढो. And then find out, and then find out, and then find out. मतलब जानबूझ के मैं error निकाल के at a higher level error निकाल के मैं उसको कम marks देती थी. But then also now these two children, they get 99.8, 99.6. But these children are those children who have studied every part of the chapter very nicely. So you have to time management बहुत करना पड़ेगा. You have to find out time so that you read. Now see, I will not say that it is so very easy also, but it is not impossible also. Now for both of you, Vignesh is not serious, number 1. And such है? Huh? Who's serious? How much did you get in the last exam? Tell me. नहीं, marks बताओ.
Bio.
Bio का याद नहीं है.
सिर्फ ज्योग्राफी मैथ्स है.
Maths का याद
कम है.
हाँ, तो you can put in effort. No, see, most of the time you are able to answer, but sometimes you are absolutely blank. Blank cup when you are not listening, when you are not understanding. And this one you can really do it. You can 80 on 80 you can get.
बदलना कितना बार तो 70 आता है. टर्म में भी 70 है.
नहीं तो तुम्हारे कॉन्सेप्ट्स कहीं ना कहीं you take it lightly. 70 क्यों आता है?
हम टर्म में मैंने किंगडम क्लासिफिकेशन छोड़ा था. टर्म 2 में मैंने इकोनॉमिक इंपॉर्टेंस छोड़ा था. और
उसमें से ज्यादा क्वेश्चंस आ गए.
हेल्थ एंड हाइजीन में लास्ट वाला वेस्ट वो छोड़ा था. पता नहीं इसके 10 मार्क कहां से कटे मैंने पता नहीं. बाकी सब मैंने पढ़ के गया था अच्छे तरीके से. ये डाइजेस्टिव सिस्टम, रेस्पिरेटरी सिस्टम और बाकी जितने भी थे वो सब पढ़ के गया था. किंगडम क्लासिफिकेशन पूरा पढ़ के गया था.
अच्छा चलो. In the 9th all those chapters were there where understanding was less of understanding and more of learning was there. 10th में वैसे नहीं है except for evolution. अब evolution जो सब chapters है, once we are done here, revise करो. You don't leave out any chapter. 10th में वैसे मत करो कि I leave out 1-2 chapter. तुम छोड़ के कैसे? 9th you can take risk, चलो not a problem. But 10th you don't take that risk. Full chapter मत छोड़ो. Even if these are the self-study chapters we give, self-study में भी तो questions तो आएगा ना. So you have to plan your time, see that you read every particular chapter. Basic idea सारे chapters के अच्छा होना चाहिए, and the major chapters you have to study well. Now which of the chapters little bit you can go relaxed is here if I talk about evolution, but evolution में भी questions तो आएगा. You should know the concept, like we will study Darwinism है, Lamarckism है, उसमें general things लिखोगे तो भी marks मिलती है. But stages के नाम है etc वो तो पढ़ना नहीं पड़ेगा. In the 10th, don't take that risk of leaving a full chapter at all. हर एक chapter पढ़ना तो है. Pollution है, that little bit you can write on your own except for few things which are specific. वैसे एक आध चैप्टर थोड़ा रिलैक्स जा सकते हो, but if you want to hit that 78 and 80, you have to go through every chapter nicely. उसके लिए टाइम मैनेजमेंट बहुत इंपॉर्टेंट है. राइट फ्रॉम द बिगिनिंग अगर नहीं करोगे, इट्स डिफिकल्ट. बाय द टाइम इट इज अगस्त, यू विल बी अंडर सो मच ऑफ स्ट्रेस. एग्जाम है स्कूल का एंड फिनिशिंग पोर्शन, न्यू चैप्टर्स. So as much possible, अभी जो भी chapter खत्म हो रहा है in the month of May, try finishing at least 3-4 chapters खत्म करके रखो. Otherwise, it's going to be difficult. Now we talk about distinctions. RNA and DNA. लिखो फटाफट. Two differences we will write. DNA. The first point is composition. DNA made up of 4 nitrogenous bases: adenine, guanine, cytosine, thymine. RNA instead of thymine, uracil. RNA instead of thymine, uracil is there. DNA is double-stranded and RNA is single-stranded. So these are some differences that you know. Next is nucleosome, nucleotide. Now you know that difference. What is the difference you will write? And this is a very common question in your paper. Take that again. This is a very common question in your paper. Nucleosome, nucleotide. I'll just put a dot with this one. Nucleosome, nucleotide. What are the differences?
Nucleosome is DNA plus histone.
Packing unit of DNA molecule. Keep your answers also very specific. Nucleosome is the packing unit of the DNA molecule consisting of DNA and histones. Then nucleotide क्या है?
Consists of
repeating unit of the DNA molecule consisting of nucleotide. Repeating unit of the DNA molecule, nucleotide, consisting of nitrogenous bases.
Nitrogenous bases,
pentose sugar
and
phosphate group. Phosphate group. वो जैसे हम discuss कर रहे हैं, all these things should be remembered. Centrosome and centromere. What is the difference?
Centromere, the point of attachment of two chromatids.
नहीं, point of attachment नहीं लिखते हैं. What is centromere? It is the primary constriction. Centromere. लिख के रखो नहीं तो book में लिखो. I do not get my pen. You may write it down. Primary constriction. Centromere is what? It is the primary constriction. Primary constriction which connects the chromatids or which connects the centromeric arms of the chromosome, which connects the arms of the chromosomes. And centrosome, you know, it is the cell organelle consisting of centrioles present at the poles of the cell. Centrosome is the organelle consisting of at the poles of the cell. लिखते जाओ ये सब आंसर्स यहाँ पे. किसका बुक है ये तुम्हारा? लिखो. Some of these I told you. Centrosome क्या लिखना है? Centrosome is
point of constriction of
the primary constriction which contains or primary constriction which connects the arms of the chromosome. Connects arms of the chromosome. Pencil से लिखो नहीं तो. And the other side is centrosome. The organelle which contains centriole. Organelle that contains centriole. Present at the poles of the cell. Present at the poles of the cell. So this is. Can you get that scale? Present at the poles of the cell. Now, what next? Haploid diploid, very common to get in your paper. Haploid diploid क्या लिखना है?
1/2 sets of chromosome.
Haploid is 2 half sets of the chromosome. Haploid, I'm sorry, 1/2 sets, and diploid 2 half sets of the chromosome. A cell containing 2 half sets of the chromosome is diploid. A cell containing 2 half sets. वहाँ पे key terms लिख के रखो. What should you write? Cell containing 2 half sets of chromosome, diploid. Cell containing 1/2 set, haploid. 1/2 set होगा तो haploid. What next? Now this one. Write out the full form, you know. Odd one out, find out the odd one out here. Complete this. Complete this one. Number 5, you complete. Number E, sub number 1, did we do the last day? We had finished. Number 3, did we do the last day? 3 किया था? अभी question number 5 complete करो. Question number 3 complete करो. 5 complete करो. Question number 3 करो. Number 4, similar question I had given you the other day as class work. किया ना? That is done. Question number 4 जो मैंने दिया था that was similar. अभी question number 5, question number 3, back there, question number 6, question number 7. Complete this. Presentation check karo. Now that you are in the 10th, presentation matters a lot. How you write your answers. Don't be very casual in the way you write. Don't be very casual in the way you write. In the book itself, if you are writing, there also write down the key terms nice and key. Check. Every answer you should be able to write so nice that whoever is correcting your paper easily impressed and gives you marks. डाउट है पूछो.
फर्स्ट
थर्ड वाले क्वेश्चन में फर्स्ट सेंट्रियोल होगा कि सेंट्रोल होगा?
Centriole. It's marking exactly at the centriole. See there? Exactly at that rod. Any doubt you have, you clarify. Vignesh.
The fifth question. The diagram given below represents a certain phenomenon which occurs during meiosis. Name and explain the phenomenon by using the terms homologous chromosomes, chromatids, and crossing over.
Answer?
Fifth question.
This one. Name and explain the diagram. Okay. A certain phenomenon which occurs. Name and explain the phenomenon. Crossing over. No? The phenomenon is crossing over by using the term homologous chromosomes, chromatid, crossing over.
This is variation.
No, phenomenon is crossing over. The diagram given below represents a certain phenomenon which occurs during meiosis. Name and explain the phenomenon by using these terms. So what are the term? What is the explanation? The explanation is the diagram represents what? The diagram represents crossing over. Name and explain. Name, crossing over. Explain. So crossing over you have to write using this terms. Write down. Explain. Crossing over is the process. Crossing over is the process in which crossing over is the crossing over is the process. It's a process. Crossing over is the process or phenomena also actually you can write here. Process in which chromatids, sister chromatids, in which sister chromatids of sister chromatids of homologous chromosomes. Sister chromatids of homologous chromosomes overlap and exchange chromosomal segments. Exchange chromosomal segments resulting in formation of recombinants. Formation of recombinants. वो जो exchange वाला chromosome होता है उसको बोलते हैं recombinants. Resulting in formation of recombinants. What does it give rise to? Variation. Continue करो, Vignesh हमारे book में.
fifth का भी करना है, fifth.
5 I told you this 5. This 5. This 5. Okay, this you have done very good. Number 3 karo. Number 3, this one and this one. Complete all the exercises. Do you have your volume with you? देखते हैं previous board questions क्या था इस chapter का. By the time you finish this fast. वॉल्यूम के जितने भी क्वेश्चंस हैं सारे क्वेश्चन सॉल्व करना है फ्रॉम द चैप्टर. सी नाउ एज आई टोल्ड यू, इफ यू आर एबल टू मैनेज going through the chapter seriously, not nicely, not only superficially एक बार देख लिया, not that. Don't be overconfident क्या ये तो मुझे आता है. No, go through nicely. क्या ये मुझे सच में अच्छी तरह से आता है. Every particular chapter minimum 5 times अच्छी तरह से पढ़ना है. Solve maximum questions possible. इसके बाद भी अगर कोई और sample papers तो solve करना ही है later. After the month of August you start that. And apart from that, if any other reference book. Now the reference book is, I have the other textbook also, which I generally give to my students. So in the form of some PDF, I can give you those questions. Maybe to one of you I can share, and then you share it among all four of you now, 5 of you. So you share, and then constantly keep solving that. जो भी doubts है, you clarify. The more questions you solve, you are more or less confident कि वही सारे questions घूम फिर के वही आता है.
Name the cell organ that forms the aster. So shall I write centriole or centrosome?
Cell organelle, centrosome. Centriole is a protein. Cell organelle is centrosome. करो, complete करो.
And what will be the second part of it? Will it be chromatid or chromosomes?
Chromatid. Chromatid.
हमें 50 वाला करना है. Name the shape, prior to the stage and or
वहाँ पे पेंसिल से लिखो ना.
हम ड्रॉ करें.
हाँ. Draw a neat diagram and show me.
So I'm already drawn.
Okay.
Yeah.
Fifth, just now I gave you the answer.
And what type of a cell is A? Is it animal?
Which one?
Yes.
It is an animal cell.
Sir, name the cell organelle that forms ashes centrosome.
Centrosome. Central क्यों नहीं लिख सकते? Because central is the protein. What type of protein is the esters made up of? What type of protein? What the estral threads or the esters are made up of? What type of protein? In the chapter study, microfilaments. Microfilaments. Right from the first exam, put in your effort. So don't get up only before the pre-boards. Right from the first exam, target रखते चलो कि 20 mark का exam है, तो nothing less than 80. 40 mark का exam है, तो 35 plus 38 तो मिलना ही है. So keep that that target every time. Keep that goal every time. Then only you will be able to make it. If those set of questions are over, just few more questions from the previous thing on pencil or pen if you have extra. Then what will you write there? I'll just mark the questions, then complete this. I'm just marking some of the most important questions, but you will go through all. Try solving all as homework later.
Now, how shall we draw interface?
Interface, nuclear membrane should be there. Chromatin fibers extensively coiled.
Because their ruler names the stage that should precede the earliest of these stages. So that would be interface.
The preceding stage. No, the preceding stage is yes, interface. So how will you draw the interface? It will be a cell with the nuclear membrane intact. It's an animal cell. Nuclear membrane is intact. On one end is the centrosome. And the chromatin is extensively coiled. The chromatin fibers extensively coiled. This is the stage that you draw. Just before prophase, interface. Here they are naming it as which stage? Interface only. See, interface, this stage. See those questions that I have marked in the volume, previous board questions. I have marked some important 5, 6. But every question there, you will have to go through. Bring your volume every day to class. Chapter wise, as we complete the chapter, from there the previous board questions, some I will be solving. But you will go through every question. Wherever there is a doubt, the next day you will come and ask. विघ्नेश हो गया ये सारे कंप्लीट करो फास्ट
And the cell component visible only during cell division. Chromosome.
Same as same.
forms of the same gene occupying the same position on homologous chromosomes. Option chromatids, B alleles, C.
What is it? What is it? Read again.
The alternative forms of the same gene occupying the same position on homologous chromosome. A. Chromatids, B. Alleles, C. Autosomes, and D. Centromeres.
Alleles.
What are alleles?
I'll tell you. Alleles are what? In the next chapter, genetics, you're going to actually learn that. Now, say suppose I talk about a pair of chromosomes. This is one. This is another. I already told you, that the 6th chromosome of the father, 6th chromosome of the mother coming together, etc., etc. Now, say suppose I understand this is one gene, this is one particular segment. Say suppose this is one particular segment. I'm saying that this is a gene, and this gene is responsible for the eye color, maybe. Then, what is the gene here in the mother's chromosome? It is maybe capital B. In the father's chromosome, maybe it is small b. Now, what do I write? B and B, what are these? This is based on the chemical composition, what is present here. This is based on the chemical composition, what is present here. The chemical composition of this gene is as such. Remember what was the definition of gene? Encodes a protein for a character. So now this chemical composition encodes a protein which is responsible for the brown color. This gene encodes, this gene in the father's chromosome encodes a protein which is responsible for the blue color. Encodes a protein for a character. Remember that. So now, are these two genes in the father's and the mother's chromosome bringing about the same character? Yes, eye color. But are these two forms of the gene the same? No. So I would draw the chromosome something like this. I would say this is capital B, this is small b. What are these B and B? B and B are variable forms of one single gene, and this gene is regulating the eye color. So I would say B and B, and there are many more, are alleles. What are alleles? One particular gene can have many forms. तो एक पेयर में two different forms हो ही सकता है. Now that you have a pair of chromosomes, maybe in your 9th chromosome there is one gene which is regulating your hair texture. In the 10th chromosome there is one gene regulating your height. But talk about the two genes in the two chromosomes. The chemical composition in both the chromosomes is not the same. अब उसमें भी, if I talk about a person, now I am this. I have got in one of my chromosome, एक chromosome में allele for the eye color is brown and another chromosome में allele for the eye color is blue. तो alleles का मतलब क्या हुआ? Alternate forms of the gene. जो, but both of them will regulate the same character, eye color ही. Then what is my eye color supposed to be? कैसे हो सकता है? दो अलग अलग है. How can be my eye color? What will be my eye color? My eye color is brown. क्योंकि ये दोनों में से जो सबसे पावरफुल एंड डोमिनेंट होता है वही एक्सप्रेस होता है हमारे बॉडी में. This we will learn in detail in the next chapter, genetics. So as of now, understand the segment of the chromosome that we have in our pair. उसमें दोनों में same composition, chemical composition हो भी सकता है, नहीं भी हो सकता है. अगर नहीं हुआ तो वो वेरिएबल फॉर्म्स को क्या बोलेंगे? ऑल्टरनेट फॉर्म्स को क्या बोलेंगे? एलील्स. What are alleles? The alternate forms of a gene. Complete करो fast.
इसका डीएनए एक से
Now, how many chromosomes will each daughter cell have after the completion of the above division? 16.
Identify whether it is a plant or an animal cell. Animal cell. Name the stage depicted in the diagram. Anaphase. Name 16. Okay, 16 you are saying. Anaphase. Okay. Identify the stage. Anaphase. Is it a plant or an animal cell? Draw a niche label diagram which follows the how many chromosomes will each daughter cell have. What is the confusion here?
So how many chromosomes? 2 or 4?
देखो डायग्राम देख के मुझे बताओ, तुम खुद बताओ. I have already explained you this concept so many times. How many daughter cells will be formed? 2. हर एक daughter cell में कितना क्रोमोसोम जाएगा?
2 ही होना चाहिए.
अरे इसमें
तो 4 sister chromatids are there, so they will again join.
This particular cell, when it divides, this is going to be one cell, and this is going to be another cell. तो ये चारों क्रोमोसोम इस तरफ जाएगा.
हाँ, वो chromatid है ना? Chromatid है chromosome.
This 2 chromatid makes 1 chromosome. तो ये पूरा 4 इस तरफ जाएगा, ये पूरा 4 इस तरफ आएगा. The daughter cells will have 4 chromosomes. What are the parts of a chromosome? A chromosome has got 2 arms and 1 centromere. Complete करो. Vignesh एक साथ discuss करो. Question, see where there is a doubt, you clarify your doubts.
Strains, huh? Pure and hybrid strains definition. What are pure and hybrid
Strains of क्या लिखा है?
पता नहीं सर, वैसे लेकिन 5th question pure and hybrid strains bracket में लिखा definition. I guess differentiate करना.
Pure and hybrid strains. Pure and hybrid strains of any organism. Say suppose. Now, what is the meaning of pure strain? Pure strain comes from the concept. Again, we are talking about the genes and alleles. Okay. Now, say suppose I talk about pure and hybrid. Pure, I have to use one example. With an example only, you will be able to understand. What is the meaning of a pure strain, pure breed, say suppose, a pure breed of an organism? So I would say that a pea plant, let me just take these examples which you are going to learn later. A tall pea plant with another tall pea plant. Same type. What is the first generation plants you get? Tall. Again I cross this with tall. What is the second generation you get? Again tall. This is called pure breed. So what is the definition of pure breed? Pure breed means pure breed of a plant, pure breed of an animal. What is the meaning? When two plants or when plants of same traits are crossed, generation after generation, the same trait is followed. This is called pure breed. Pure breed. These are all pure breeds. Then what is a hybrid? Hybrid means a tall plant I cross with a dwarf plant. Just a contrasting item. Then what is this plant? Yes, it will be tall, but it will contain the allele for dwarfness also. It will contain the allele for dwarfness also. तो इसको बोलेंगे, listen here, इसको बोलेंगे hybrid. This is a hybrid. Understood? So what is the meaning of hybrid? An organism, a plant or an animal which contains two contrasting traits. Take that question, I will give you that answer also there. In the next class, I will give this answer. What is the basic answer that you should write for the pure breed? Again, this goes into the concept of genetics a little bit. We have not studied genetics actually, but I'll give you the answer later. When you study the chapter, you will be able to correlate better. Go for a break, and after the break, we'll continue again. Yes. They have not come, you should be a loss for them.
भाई इधर बैठ भाई सुबह से मैंने टिफिन खाया नहीं है
हाँ तू बैठ आराम से स्टेज जाने के
15 रुपया सिक्का मेरे जमा करना है भाई को नोट है ही नहीं दो
मुझे क्या पता मेरी पेंशन
आइए तुम्हें एक टमाटर बहुत है
बायो थी बायो पढ़ा रहे हैं
हाँ
एक दो तीन चार पाँच छह सात एक दो तीन चार पाँच छह एक दो तीन छह 12 13 14 15 हो गया भाई घर जाने का भाई तो बस 1000 रुपये 30 रुपया हो गया तो लॉटरी
जरा के बैठा
हाँ
कुछ दिखा नहीं
इसे गिरेगा नहीं What? Hey, phone का से? Oh, yeah. Forward forms. ये अलग मैसेज कर रहे हैं.
कौन
हाँ ये था.
हट गई
Forward forms. कुछ भी लिखते हो भाई. कुछ भी बोल दिए कुछ भी लिख दिए.
भैया मेरे फोन डाल दे देखो
अब ब्रेक है. ब्रेक क्या करें?
रियल दो मिनट में दो बार मीटिंग को बाहर करते हैं, तो चीटिंग क्यों होती है भाई? इसलिए आप लोगों को भरोसा दिलवा दिया कि भाई हमारे पास कुछ नहीं है, यूजर से मैंने चाह रहे हैं आप की वजह से बातें हैं जैसे इस चक्कर में यहाँ पर इसलिए एडिकेट और होशियार को बाहर कर दिया, लेकिन रियल दो मिनट में दो बार मीटिंग को बाहर करते हैं, तो चीटिंग क्यों होती है भाई? पिछले 3 साल से आई थिंक
बच्चों का ना अंदर
बता क्या है कि बच्चे लिखोगे बात कर भैया चार्ज